we found that about half of all biomarkers that were reported were actually related to quality issues. This is really one of the things that we guess is an important topic, so we really need to be sure about the QC of our samples. And another point that we think can really accelerate this biomarker transition is to not work in too small studies. So many of the published biomarker studies are underpowered. And what we are emphasizing is really to look at larger cohorts of several thousands of samples.
Welcome to the Personalized Medicine Podcast. This is the place where scientists, clinicians, and entrepreneurs discuss the progress of this rapidly developing field. I am your host, Alexander Yahensky. Let's start. 3, 2, 1, and we are live. Welcome to the next episode of the Personalized Medicine Podcast. If you have been following us for some time, you know that we talk a lot about biomarkers on this show. Proteomics is one of the fields which has seen an enormous boom in terms of technological advancement over the last decades. Improvements in mass spectrometry throughput, automated sample preparation, and growing impact of AI on data analysis brought proteomics to the spotlight of precision medicine. I'm very happy to welcome on our show the expert who is driving the future of proteomics further forward, Dr. Sophia Doll. Sophia is the co-founder and chief business officer at OmicEra Diagnostics, a Munich-based startup that is leveraging mass spectrometry and AI to assess biomarkers of various human diseases. Sophia did her PhD at the Max Planck Institute for Biochemistry in Munich, where she studied translational protein biomarkers of cancer in the lab of Matthias Mann. She conducted part of her graduate research at the University of California in San Francisco and worked for Genentech, where she analyzed epigenetic and proteomic alterations in lymphoma. In 2019, Sophia co-founded OmicEra and has been serving as its chief business officer since. Sophia, thank you so much for accepting my invitation and welcome on our show.
Sure, thank you very much. Thank you very much for the nice intro and having me on your podcast.
Great. So let's start right away and let's start with your story. Can you tell us a little bit more about your PhD and postdoc research and what motivated you to start a company in the proteomics space?
Sure, I'd be happy to. So my PhD was actually a real journey. So as you were just saying, I did my PhD in Munich and San Francisco at 3 different institutions. I started to work at the biotech company Genentech and then moved on to the University of California, San Francisco, and then went to the Max Planck Institute in Matthias Mann's lab. So you can imagine as a PhD student, this was really great, was coupled to quite some travels, and I also learned a lot and explored the interconnections between industry and academia. So this was really, really great. My PhD was not only a journey because of the geographically locations, but also because I investigated system-wide alterations in cancer at several biological but also cellular level using mass spectrometry-based proteomics. So starting from the nucleus of the cell, I explored epigenetic changes in lymphoma and then moved out of the cell of the nucleus of the cell to the cellular level, where I analyzed phosphorylation signaling alterations in glioblastoma cell lines. And then I moved from more biological to translational cancer proteomics. And what I did here is I was working with human cancer tissues. So this was extremely exciting to me because to finally get to work with real clinical human tissue. This is something that I thought was quite exciting. And what I did here is I optimized a MS-based proteomics workflow for the rapid screening of all kind of clinical tissue samplings. enabling that way high throughput analysis of hundreds of samples with great depth. So this was a quite important step because this is exactly what you need in the clinic. So you need to be fast, so you need high throughput, high turnaround time, but also you need to be very reproducible. And this is what we achieved with working on that sample preparation workflow. And with that in place, this actually brings me to, I would say, my highlight of the PhD and something that really motivated me to actually found OmicEra Diagnostics. This is about a study where we showed for the first time that with mass spectrometry-based proteomics, that remains a very complex but extremely powerful tool, that we can, with that, identify alternative novel therapeutic targets in end-stage cancer patients. So this was a very motivating story for me, a study for me, because here together with physicians, we really were investigating, looking for these alternative treatment strategies. And it's with mass spec-based proteomics that we could actually identify an alternative treatment and manage to convince the tumor boards, but also insurances, to pay for the treatment that we recommended based on our technology. So if I say this was kind of a key from bench to bed moment for me, and really excited me because I'm burning to see how MS-based proteomics can really be used for clinical relevant questions and be directly applied in a clinical context and have a direct impact. And that's exactly what we aim to do at OmicEra Diagnostics. So this is kind of my journey through my PhD and postdoc, and that finally then also motivated me to start my own company in the proteomic space.
Fantastic. Sounds really exciting. Sophia, it would be really great if you can tell us a little bit more about OmicEra, the beginnings of your company, perhaps. Who were your co-founders, how you found them, and what were the first challenges that you had to overcome when you were establishing this company and taking it off the ground?
So it's during my postdoc end of PhD that I actually met my two co-founders, so Philipp Geyer and Sebastian Winter. We were sitting in the same office. Our office was called the clinical office. And we worked extremely well together. We had a lot of fun at work, and also the three of us had very different backgrounds. We kind of were very complementary. So Philip, during our PhD, we called him king of plasma proteomics because he knows every protein by heart in plasma you can find. Sebastian is very techy, very smart. He invented also the EasyTag for multiplex protein quantification. And myself, I had, as I was just saying, more of a cancer proteomics background and broad industry experience. And all three of us during our postdocs were working on clinical relevant project and all shared that vision of transforming this mass spectrometry based technology from being a tool for specialized laboratories, as we used in our PhD and postdocs in academia, to really transform it to a powerful technology for translational proteomics. So we really wanted to see this technology also being used in cutting-edge clinics and eventually deliver personalized treatment options, as I was just referring in the previous question. So together, we decided to quit our postdoc and start our own journey, our new era, and found OmicEra Diagnostics. We secured fundings from angels investors and started to look for lab space. And you were also referring earlier to what the main challenges were, right? Especially at the beginnings of OmicEra. And one that was quite surprisingly, but one of the biggest challenges we had was to find lab space. So at first we could only get 2 small rooms, about 20 square meters or so, at the Startup Center for Biotechnology, the IZB, which is very close to the Max Planck Institute. But obviously you can imagine that very quickly it was not enough. It became too small, too fast. And there the challenge started, right? We called many companies and real estate agent, and we're never lucky. I mean, also we're in Martinsried, which is kind of a biohub for biotechnology in Munich. It was quite challenging, but at the end, yeah, we got lucky and found an office that we could completely renovate it. Our landlord was really nice. She was super excited about the science we are doing and trusted us and really gave us this opportunity to completely build in a new lab in this quite large space. So we have 250 square meters and room to grow. And I'm always looking for talent. So in case of someone of the listeners is interested in joining, please don't hesitate to reach out to us. We're always looking for great talents to join us.
Fantastic. Yeah, sounds as such a great story. And obviously you had to overcome so many challenges just starting the company and finding the lab space. Who would think that this would be a big bottleneck, but it is a tricky issue. And great to see that you found some support and could actually build your own lab from scratch. I think it's extremely exciting.
Yes.
Yeah, and for all of our proteomics experts in the audience, we'll make sure to add Sophia's contact details in the show notes. So make sure to check OmicEra website and reach out to Sophia in case you would like to join their team. Perfect. Sophia, what I'm trying also to understand, and it would be great if you could share with our audience, what exactly is that you're doing with OmicEra? What type of services do you provide, and how you can accelerate the transition to discovery and reliable assessment of biomarkers for various diseases?
Yeah, absolutely. So at OmicEra, we aim to improve health by proteomics. We thrive to see our MS-based proteomics technology platform to be actually implemented in the clinic and revolutionize current medical diagnostics and basically, yes, to improve health of every individual. And what we provide currently at OmicEra is our MS-based proteomics technology platform as a service for the discovery of novel or more reliable biomarkers for early disease diagnosis. And as you know, over the last years, only very few novel biomarkers were actually approved, and most of the ones we're currently using are older than 20 years. But we all know that it's really key to detect diseases as early as possible. So really emphasizing this urgent need to uncover new biomarker. And this is exactly what we provide at OmicEra. So with our high-end MS-based platform, we really want to support our clients, so which are mostly pharma biotech companies, to uncover biomarkers in clinical studies. We basically work as a CRO and provide this OmicEra platform. And maybe to go a bit more into detail, because you were also asking what exactly is our service, what is our product. So what our platform is and what we offer is complete proteome analysis from sample input to data output. We work with all kind of body fluids, so everything you can think of, from plasma, CSF, urine, saliva, you name it, as well as tissues. So with tissue, we work with fresh frozen tissue, but also FFPE sample, also with very low input, such as laser capture microdissected tissues. So we have a very wide range of different sample types we can work with at OmicEra. We process all samples with our automated sample preparation pipeline, measure the samples on our timsTOF Pro instrument here in-house. And after data interpretation, what's important to us is not only to report a list of proteins to our clients, but to really get the most out of our clients' data. So what we're aiming here is really to maximize the insight into the data. And for that, so we perform AI-based bioinformatics analysis of the samples by really integrating the information with our internal data bank. And if we also get clinical information from clients on top, this even further maximizes the insight of these proteomics data. So it's very important for us to try through different correlation analysis with our know-how to really kind of get the most out of the data. And this is, yes, what we provide to our clients.
Yeah, fantastic. I think this back-to-back analysis is extremely important, like given a lot of variability that can come from sample handling and processing and measuring and then analyzing the data, and just to have everything under one umbrella certainly helps. And Sophia, I also would like to discuss this last point that you mentioned on bioinformatics analysis. I know that you have recently released this OmicEra platform. So perhaps you can tell our audience a little bit about what it actually is, what it can do, and how it can help in analyzing those large datasets of proteomics data.
Yes, so indeed, we very recently released our Theomiclearn platform. So this is a project that was led by our bioinformatics team, especially by Maximilian Strauss, who some of you might also know. He is the main developer of the open source AlphaPep search engine, and we are very fortunate to have him also here at OmicEra Diagnostics. And Max, together with his bioinformatics team, just released this Theomiclearn platform that is an open source and web-based machine learning tool. So what Theomiclearn does is it uses the latest advances in the Python ML ecosystem and really helps scientists that are not experts in bioinformatics or machine learning to very easily analyze their data. So most of them are usually proteomics data, but you can upload any kind of omic data, and allows them to use state-of-the-art machine learning algorithm for classification and for really gaining insights into their data sets without requiring any very specific programming skills. So it's very easy to follow and ready to use. As said, it is open source, so you can check it out at omiclearn.com and just start using it. It's very just a drag and drop system. It's very easy. We usually upload, for example, MaxQuant or AlphaPep or any kind of other search engine output data, and then you can start to play around and analyze your data. So yeah, that's basically Omiclearn that enables rapid exploration of the suitability of various ML algorithms for your experimental data set of any kind of omic data set.
We are doing this show for you, and your feedback is very important for us. So if you have any suggestions or comments, would like us to cover a specific topic or recommend a guest, please write us an email to team@pmedcast.com. Or you can reach out to us on LinkedIn, Twitter, or Facebook. Just type in Personalized Medicine Podcast and you will find us there. To download the show notes for this episode, visit our website, pmedcast.com. It's pmedcast.com. The show notes include guest bios, links to their most notable work, and recommendations for additional reads on the topic of the episode. Make sure to check them out. And don't miss the next episode. Subscribe to our podcast on your favorite podcasting platform. Give us a rating and leave a comment. It will help us make this show better. And now, let's get back to the interview. One more point that I want to discuss with you, Sophia, is on the proteomics as a whole. You mentioned already that despite a lot of progress in improving mass spec throughput in general, just in our understanding of the proteomes of various tissues and plasma, for example, we still translated relatively few of potential biomarkers that are published in scientific literature into clinics. So in your understanding, why is that the case and what can we do to accelerate that transition?
Yeah, sure. So as you were saying, there are not so many biomarkers that actually made it into the clinics, and most of the ones we use are quite old, so more than 20 years or so. And so what we've done is a literature research where we found that about half of all biomarkers that were reported were actually related to quality issues. So this is really one of the things that we guess is an important topic. So we really need to be sure about the QC of our samples before reporting any biomarker issues, because we've seen in many studies that if you collect samples at different location, you might see important outliers, but that are just related to the way how samples are handled, or if you have, for example, hemolysis or things like that. And so to avoid that in the future, to report biomarkers that are actually just related to pre-analytical variation, we have developed panels to really always check for the quality of each sample and only report biomarkers that we are 100% sure are not related to any potential sample handling issues. And another point that we think can really accelerate this biomarker transition is to not work in too small studies. So many of the published biomarker studies are underpowered. They have too small cohorts. And what we are emphasizing is really to look at larger cohorts of several thousands of samples. This is the rectangular strategy that our co-founder Philip Gaia actually published in Molecular System Biology in a review called Revisiting Biomarker Discovery, where he really emphasizes to look in large studies instead of looking in a small cohort and then following up the targets with a targeted approach. Here it's really about looking in large cohorts in an unbiased way, what would be potential biomarkers that seem to be specific to one disease phenotype, and then validate it in a second larger study. So this is what we think could really help to accelerate biomarker transitions into the clinic.
Perfect. Yeah, let's hope that there will be more and more studies with significant n size and also with a more rigorous sample preparation, because that's the key, as you said, in terms of reproducibility and then getting the actual insights out of that proteomic data. Essentially, if you look at application spectrum of proteomics, it has been increasing over the time as well. And obviously now in the times of COVID pandemic, that's another field, infectious diseases, where proteomics is playing an important role. And I know that together with your colleagues, you have recently published a paper on the proteome trajectories in the plasma of COVID patients. So perhaps you can tell us a little bit more about that research and which insights could you draw from your analysis there?
Yes, so this was actually an exciting study. It was led by Philip Gaia, that was just referring to, co-founder CSO of OmicEra Diagnostics. And he worked together with the LMU Klinikum Großhadern, which is one of the largest hospitals of Bavaria. It's here in Munich. And in this study, what we aimed for is to identify proteomic changes in blood serum that would help to predict which individual will get really sick from COVID infections from those who will have a more milder disease progression. And this is extremely important to be able to predict as early as possible, because you may have also heard of people having very severe disease progressions, but some a more milder disease progression. And if we could somehow predict that a bit better, this would be extremely important for better management of intensive care units and avoid that they get overfilled. So in this study, what we did is we applied our OmicEra technology platform to uncover biomarkers for COVID-19. And as you were just saying, in a longitudinal study. So what we mean with that is it was a study where we collected at several time points from a patient over the disease progression blood samples. And that was really nice. So we could actually really follow the trajectories of protein change and over the course of the disease infection. And we did that for patients that were actually COVID positive, so based on the PCR test that was positive, but also of patients that had very similar symptoms to COVID, but were actually negative, revealed by the PCR test. And what we found is that in about a quarter of all proteins, that they were changing significantly up in COVID infection. And while proteins, for example, of the innate immune system decreased early in the time course, other proteins such as coagulation proteins and proteins of lipid homeostasis increased quite a lot over the course of the disease. And this was very interesting to observe these protein changes during the disease infection. And what was even more interesting is that we also uncovered a potential prospective biomarker for COVID-19 survival. And the study we published very recently is actually only the first part of the entire story. We're working on a follow-up second study to actually validate these observations. And this second COVID serum study is, to our knowledge, the largest serum proteomic study that will be published to date. We enroll about 3,000 patients and also have clinical information for each of the sample, and are extremely excited about that because it's not always easy to get access to those data that are sometimes not so well documented, but they will give us so much more power in actually analyzing the data and really increase our insight into the biology of the COVID-19 infection reflected on the serum proteome. So yeah, we hope that overall we somehow will be able to contribute to a better understanding to this ongoing pandemic.
Yeah, fantastic. And it seems like there is no limit to what proteomics can do, right? It's really important to understand, especially in case of COVID, why some patients are actually not responding so severely to the disease while others are really suffering a lot. And let's hope that proteomics can shed some light on that and will help us combating this dreadful pandemic. Sophia, in the end, we always like to ask the question about the future. So if we look at the field of proteomics in general, biomarkers and mass spec specifically, what are the major developments that you see happening in the next 10 years or you would like to see happen? And how could they help us bring the entire biomarker space and precision medicine forward?
Yes, so there have been already so many improvements, but of course, many more to come. And where I see the most developments probably coming from is on the technology side. So our mass specs are getting more and more robust, which is really essential that you have a mass spec that you do not need to clean all the time because you need continuous results over a long period of time, especially if you analyze large clinical studies. So this is very important, and we do really already see a very strong change. Instruments are getting more and more sensitive. This is also very important, and I hope that there will be a continuous growth over the next years. Instruments are also getting smaller in size, also important to be able to enter into the clinic. And something that we also see, and that I really hope will happen even further over the next years, is that the prices of the instrument need to decrease. We've seen this in the genomics field, right? I mean, 10 years ago, it was quite expensive to get a whole genome sequenced. Nowadays, it's very affordable, and the same thing will happen for proteomics technology. I also hope that in the next 10 years, I mean, a mass spec will be very affordable. And all these technological advances, together with AI and ML approaches, as well as automation of the sample processing to enable higher throughput, are all important developments so that proteomics, MS-based proteomics, will actually be able to support clinical trials and clinical decisions. So these are the improvements I really hope to see very soon.
Perfect. Yeah, let's hope all of this will happen, and it looks like it's happening already. As you said, the technology is improving, and I think this parallel with genomics is really spot on. Like if you look back at the cost of, I don't know, Illumina sequencers 10 years ago, 15 years ago, it was a completely different price range that it is today, and then the cost of the sequencing of the entire genome went down dramatically. And yeah, let's hope the same will happen to proteomic screens of plasma, for example. Great. Sophia, you've obviously done very interesting, many people will say very bold transition from science to starting your own company. I guess there are a lot of scientists who are listening to this podcast also early in their career who are thinking about starting their own company or joining a startup. Which advice would you give to those people from your personal experience? Should they do it, and why?
Do it. I mean, that's probably what I can tell everybody. It's really worth it, and maybe especially as a woman in science. So to all women scientists who might listen to that podcast, I can only say always think big, take a risk, and most of all, believe in yourself. Don't be shy about your thoughts and dreams. It's really worth it to pursue. And if you find the right team, which is crucial, it's very important to have a team that you can trust, that you work well together. And if you have a great vision, well, then just go for it. I mean, it's really worth it, and just dare it.
Great. So let's hope a lot of people in our audience will apply it. And Sophia, before I let you go, can you let our audience know where can they find you online in case they want to reach out to you?
Absolutely. So yeah, please feel free to reach out directly via email. My email is doll, DOLL @OmicEra.com, or just simply write me via LinkedIn, or you can also find us on our website, OmicEra.com. And yeah, if we're always open for partnerships. So if you think that somehow whatever you're doing could be complementary to OmicEra, we're always very open and really believe in partnerships to bring the technology together forward. So feel free to reach out. And also, if you think that OmicEra is interesting and are currently looking for a new job opportunity, also please reach out to us. We are always looking for talent to join our fun and amazing team.
Perfect. Sophia, thank you so much. It was a pleasure to have you on the podcast. Thank you for sharing information about your company, discussing proteomics with us. It was extremely insightful, and I'm sure our audience will love this episode.
Thank you very much for having me. It was a pleasure. This was fun. Thanks a lot.
Thank you so much for being with us today on the Personalized Medicine podcast. If you like this show and know someone who would enjoy it too, please share this podcast with them. And don't miss the next episode yourself. Subscribe to the Personalized Medicine podcast on your favorite podcasting app. You can find us on Apple Podcasts, Spotify, Pocket Casts, Stitcher, iHeartRadio, and many, many more. Please rate us there and leave a comment. That helps us to grow and deliver the best experience to you. To access the show notes for this episode, visit our website, pmedcast.com. It's pmedcast.com. And engage with us on social media, where we regularly share the news and exciting content on personalized medicine. You can find us on Twitter, LinkedIn, and Facebook just by typing in Personalized Medicine Podcast. Or use our handle, pmedcast. And if you have any feedback or would like to suggest a guest for the show, write us an email to team@pmedcast.com. Have a great day, and until next time.